Let's say you're curious about the function of a particular gene of interest in a specific eukaryotic organism. A good way to determine what a gene does is by looking at situations in which the normal function of that gene is altered. One way you could do this is by looking at organisms with naturally occurring mutations in that gene. But say your gene of interest doesn't have any of these mutations. How then do you observe alterations in your gene's expression? Well, there are two ways to silence your gene by limiting or completely eliminating its expression. Two powerful ways to do this are through RNA interference and CRISPR. RNA interference occurs naturally in eukaryotic cells and serves as a mechanism to limit the invasion of foreign genes and control the expression of the cell's own genes. We are able to take advantage of this naturally occurring process by injecting the double-stranded RNA of our gene of interest into the cell. What happens is the cell recognizes the injected double-stranded RNA as a foreign body and seeks to destroy it, as it evolutionarily would have done to protect itself from foreign invaders like viruses. So how does it do this? The invading double-stranded RNA is cut by the enzyme dicer, an endonuclease protein. This forms small interfering RNAs that then bind with proteins, including argonate, to form an RNA-induced silencing complex, or RISC. After one strand from the double-stranded RNA is chosen as a guide RNA, the RISC complex looks for its complementary mRNA. It then represses or degrades the translation of the mRNA. In response to viruses, prokaryotes produce two types of short RNA that form a complex with the Cas9 protein. One sequence matches the virus sequence. Cas9 is a nuclease that can cut DNA. Once again, similar to the eukaryotic analog, the guide RNA finds the target in the virus's genome, and Cas9 cuts the target DNA. When this process is modified by being introduced to eukaryotic DNA, the cell will try to repair the cut DNA. While this process is imperfect and typically results in mutations that can be advantageous to studying the gene's function, an even more useful result can be produced. If a piece of DNA carrying the desired sequence is added, it can be incorporated into the genome, allowing scientists to cut and paste genes. Which is a better method, RNAi or CRISPR? While that may depend on what you are specifically looking at, in general, I would say that there are more advantages to using CRISPR, which is probably why it is a more commonly used tool in research today than RNAi. One major, and probably obvious, advantage to CRISPR is that it edits DNA, making permanent changes in living cells, while RNA interference only makes temporary changes. Furthermore, CRISPR allows you to insert genes and target many genes at once. For those reasons, I believe CRISPR is a more powerful tool, and I would choose it to investigate my gene of interest.